Welcome and thank you for joining today's AWRI webinar. My name is Robin Dixon and I'm the Senior Viticulturist, or one of the Senior Viticulturists at the AWRI. Before we begin, I'd like to start by acknowledging the traditional custodians of the land on which we meet today and pay my respects to their elders past and present. I extend that respect to Aboriginal and Torres Strait Islander people here today. In this session, we'll look at quantitative methods to detect and estimate botrytis contamination in grapes. But before we jump in and make a start, we've got a couple of reminders to anyone who's new to AWRI webinars. So if you'd like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar, type in your question and click send to send it through. We'll be holding the Q&A session at the end of the presentation, but feel free to send in your questions at any stage. A reminder also that this session is being recorded and you'll be emailed a link to view the recording on the AWRI's YouTube channel. So for anyone who has just joined, welcome. Today's webinar topic is quantitative methods for botrytis grey mould detection and estimation in grapes. It's a great pleasure to welcome Professor Chris Steele, Professor of Viticulture at the National Wine and Grape Industry Centre at Charles Sturt University. Chris conducted his postdoctoral training in the UK, Switzerland and Australia in plant pathology and disease management. He is currently Professor of Viticulture at the National Wine and Grape Industry Centre at CSU and conducts research in viticulture and enology with a focus on the impacts of fungal organisms on wine quality. His principal research interests are on bunch rot diseases of grapes. Chris, if you're ready to make a start, I'll hand over to you.
Thank you, Robin. I'll just share my screen. Okay. So good morning, everyone, and thank you, Robin, for the introduction. So today I'm going to be talking about some of the work that my research group and some of my colleagues in other institutes have done on the detection of grey mould in grapes and estimation in grapes. So I thought I'd start off by just giving you a bit of an overview of the presentation. I'm going to briefly review botrytis, and I would imagine that anybody attending the webinar today probably knows what botrytis is already, so we're not going to go into any discussion about what botrytis is and his biology. But I will say something about the impacts on grape and wine quality. And the reason for this is that if we want to measure botrytis in some way, I mean, if we understand how it affects grape and wine quality, then possibly we can use some of those parameters as potential methods to quantify the amount of botrytis that's present in the batch of grapes. So the bulk of the presentation, I'm going to be exploring the techniques that we've developed or that we've looked at for botrytis detection and estimation. And as I go through each of these, some I'm going to concentrate on a little bit more than others because we've done more work in that area. But as I go through them, I'm going to conclude each section with a slide that says what my verdict is, what my opinion is in terms of the pros and cons of each method, because none of them are 100% perfect and none of them are completely useless. They all tell us different things. Then at the very end, I'll have a summary slide where I bring all the methods together, where I try to collate everything that I've discussed in terms of the cost, the skill set required, and so on and so forth for each of the methods. So briefly, impacts of botrytis on grape and wine quality. Well, of course, there's not just the loss of yield, but there's an impact on the quality of the grapes and the wine quality. Of course, one of the most obvious ones is formation of oxidative enzymes that leads to browning in grapes and in wine. And this is primarily due to enzymes such as lactases. And there's been a considerable amount of work on lactases. We've actually dismissed them as a good indicator because the production of lactases is quite variable amongst Botrytis isolates, but also it varies according to the environment that Botrytis is in. And that's the same for really all of these oxidative enzymes. So I'm not going to say anything about lactases, I'm just flagging it there that it's something out there. But we have looked at some of the unwanted compounds that arise in grapes due to the growth of Botrytis in the vineyard. So these are the mouldy, musty, earthy-like characters. And we've been looking at how perhaps we could use these as a way to quantify Botrytis in grapes. The other obvious impact is formation of organic acids, notably gluconic acid, but also to some extent acetic acid, but often this is coming from the growth of other organisms, and particularly acetic acid bacteria on the berry surface that occur naturally. And when the grapes are damaged, then the acetic acid bacteria take off and produce high levels of acetic acid. The other thing that Botrytis does, of course, is that it depletes some of the desirable characters that we want in grapes and wine. So we see a depletion of the fruity varietal characters. So why do we want to do this? Why do we need to measure Botrytis? Well, some of the questions you might want to ask, we think there might be Botrytis out there. Can we leave the fruit in the vine a little bit longer? So when to harvest? Or conversely, we know there's Botrytis there. How much is there? Is it too late? Should we even bother harvesting? So that quantification of Botrytis, but it needs to be done with respect to, well, if it's there, what are the implications? What are the impacts on wine quality? Factors to consider when we want to evaluate a technique or a tool or a method for measuring Botrytis, either detecting it or quantifying it, or both. Well, one of the key things, I think, for the wine industry would be the time of analysis. The fruit's about to come into the winery, how much is Botrytis there? So that time of analysis. So is it okay to have an assay that takes days? Probably not. Hours, maybe. Minutes would be the ideal sort of time frame. Then we need to think about the skill set or the training required and the access to the instrumentation that might be needed to carry out the assay. And coupled with all of this, there's the applicability for a field situation, be it the vineyard or be it at the weighbridge at the winery. And then for many of the methods that we can consider, we're not just looking at Botrytis. Sometimes we might be looking at all fungal rots. So we're really interested in that. Perhaps it doesn't matter whether it's Botrytis or something else. We're just really interested in the quality of the fruit and the impacts of fungal growth. And another really important consideration is the sample size. A lot of the work I'm going to present and the work that other people have done has been on single berries. Well, obviously you don't do that in an industry situation. You want to sample the vineyard, not a few berries. So do we look at berries, bunch, or the bin, harvest bin, and how many replicates, and what about the vineyard variability? Now, some of that I'm not really going to answer today. I'm just putting it out there as a consideration. So we're now going to move on to some of the methods that I'm going to cover. And as I said before, some of these I'm going to cover in a bit more detail than others. So first we've got visual estimations, be it with the naked eye or with some other sort of imaging type technique. We've done some work on ATR, FDIR, and I'll explain what that is in a moment. We can look at these off flavors, aromas, mushroom, earthy type aromas, these volatile organic compounds, IGC, MS. Gluconic acid is a measure. A lot of hype and interest in molecular methods. I'll say a little bit about qPCR. And then we come down to the last two that we've tended in my research to concentrate on in the last few years: measurement of the fungal steroid Agostrol, and more recently, detection of Botrytis antigens using a lateral flow device. So I'm going to go through each of these in turn. So we'll start with visual inspection. This is probably what most people do. want to know if you've got botrytis in the vineyard. You walk through the vineyard, you walk down the rows and you eyeball the bunches and randomly you select some and say, is there botrytis there or not? So it requires a little training. The issue is that it's a subjective method. So if you look at the bunch on the right, one person might say there's 60% of that bunch is affected by botrytis, and the next person might say 70%. So it's subject to error. The untrained eye might not discriminate between botrytis and other disorders, particularly on red varieties, perhaps heat damage, heat stress, bunch necrosis. Perhaps there might be confusion with other rotting organisms. So there's a degree of imprecision in this technique. In terms of the skill set, probably not much training is required, but the cost there is largely the staffing. Now the issue with botrytis, we've got this bunch here on this slide. This is a Chardonnay bunch, and you look around this bunch, you can see it's probably quite a dense canopy. We can see some shading there, some tightly packed clusters and berry splitting. The question is, how much is really there? And if you didn't look at that angle, would you actually pick up there's botrytis there? And there's some odd berries that are perhaps hidden behind the bunch, and really you need to go in and turn the bunch around and have a good look at it. So that's one of the limitations with visual estimations. We can think about other imaging techniques, so hyperspectral imaging. There's been a lot of work conducted in this area. But it's the same sort of thing. We're not seeing inside the berry. We're particularly with the reds, this is probably going to give a greater degree of accuracy to discriminate between an infected berry and what might be, heat stress or scarring on the berry. But again, we're not seeing it within the interior of the bunch. So what's my verdict on, what's my opinion on the imaging techniques? Well, they're valuable, but they are prone to errors. Some of the imaging techniques could perhaps in the future employ equipment that could be applied for a field situation. So mounting a hyperspectral camera on the back of a vehicle, driving through the vineyard. But there are some limitations in terms of the data and what's picked up, and where you've got shading in the canopy and bright day versus an overcast day. And again, with all these techniques, they don't tell you what's inside the bunch. I'll now say something about the work that we've done on ATR FDIR, which stands for Attenuated Total Reflection Fourier Transform Infrared Spectroscopy. Bit of a mouthful, that's why we say ATFTIR. It's simple to operate. The instrumentation is applicable for a benchtop type setting. Such an instrument is on the right-hand side in the top image. The way this works, when we expose the sample to infrared, this results in bending and stretching of bonds. And these bonds vibrate. And there's a characteristic vibrational frequency depending upon the functional groups that are present. So carbon to oxygen double bond, for instance. And from this, we can get a fingerprint of the sample. We only need a very small volume, and I've just brought up that little yellow square there. That's where the sample goes on the reading frame. So we're talking about microliters. Quick to do, but the data analysis is a bit more time consuming. So I'm going to show some results of a study that we completed probably about 4 or 5 years ago now. This is this ATR FTIR of detached grape berries, so individual grape berries. And you can see in this scan that we've got a tracer. It's part of the spectrum. We're not looking at the whole thing. But you can see that the red trace there is Aspergillus, and the darker blue is our control, clean berries, and the green is Botrytis. And we can see some differences there in the spectra. And so if we just hone in one part of the spectrum, we can analyse the data there to see if we can discriminate between berries. And that's exactly what we did. We did a PCA analysis that's depicted on this slide. And you can actually discriminate quite nicely between the Aspergillus infected berries and the control ones. You can also discriminate with the Botrytis ones, but it's not as good, it's not as clear cut. So the problem with this technique, it requires a bit of data handling, data analysis. The other thing that we found, and I'm not going to present those results today, is that when we looked at whole bunches as opposed to single individual infected berries, the discrimination wasn't as good. And so I see this as a limitation, because you're not going to go into the vineyard and pick individual berries. You're going to be looking at whole bunches or you know, a bin of grapes. So here's my verdict on the ATR FDIR. Benchtop instrumentation, modest cost. I don't know what the figure would be. We're probably talking maybe 10,000 perhaps. I don't know. Uses a small volume, but requires this skills in data handling analysis. We get more conclusive results with single berries rather than bunches. So probably not so good for a field situation. I'll now move on to the volatile organic compounds, and there's some examples here. These are some of the better known ones. Geosmin, 1-octene-3-ol, 1-octene-3-ol, and methyl isoboniol. Many of these are common to all fungal species, fungal spawnage in anything. It doesn't have to be grapes. And so some are not unique to Botrytis, so that's a limitation. And the figures of these ones, the figures in blue font there are the sensory perception thresholds, just for information. But when we look at grapes, berries or bunches that are infected with Botrytis, we use a GC mass spec to do this. We might get a result that looks something like this, and you can see two chromatograms here, one in blue, which is our clean material, uninfected, Botrytis free, and then the other trace in red is the berries analysed that have been infected with Botrytis, and we can see some peaks that are unique to the Botrytis infected berries. They're not present in the control material, and some of them aren't found when we have other bunch rot fungi there, like Aspergillus, Penicillium, Rhizopus, and other things. So these compounds might potentially be quantitative markers for Botrytis. So we measure the peak area, work out how much is there, then we can perhaps say how much Botrytis is present. The other thing I'll draw your attention to on this graph, just as interest, is that blue peak that disappears with the infected berries. That's actually beta-damascenone, which is of course a desirable compound that we might want in our grapes and in our wine. And you can see that that has been depleted when berries are infected with Botrytis, and possibly that beta-damascenone is going to some of these other red peaks that are there in the infected berries that are actually, some of them are unknown sesquiterpenoid compounds. So there are some compounds that are unique to Botrytis infected grapes, and this study was done with Chardonnay. So that's the other thing to take into consideration, the variety you're looking at. But some of the ones are 1,5-dimethylnaphthalene and a number of these unidentified sesquiterpenes. And so these could potentially be used as markers for Botrytis contamination and perhaps a quantitative marker. So what's my opinion about the VOCs, the volatile organic compounds? Well, we do require at the moment sophisticated analytical equipment, GC mass spec. We need someone with the capability to run the instrumentation, but some of these compounds are specific for Botrytis. So the technique has potential for specificity just to pick up Botrytis only. The analysis time is still not down to minutes or hours. We're talking about days. At the moment, we need to do this in the laboratory, but maybe in the future, some sort of handheld mobile GC mass spec device might be possible, where we can analyse the atmosphere of the vineyard, go along the row and perhaps say, yes, we've got some of these compounds at this part of the vineyard, turn the harvester off at these panels and then turn it back on at the next panel. So that's perhaps something for the future. Now talk about gluconic acid, and I'm aware that lots of people do measure gluconic acid in the industry. And of course, the ratio of glycerol to gluconic acid has been suggested as an indicator of whether we've got grey mould or noble rot in late harvested fruit. And the higher the amount of gluconic acid, then the less desirable the fruit is, the more spoiled it is. So there are various commercially available enzyme-based kits for measuring gluconic acid. These are pretty simple to use, easy to use. They come with a set of instructions. If you follow the instructions carefully, don't mix up your reagents in the kit, then they work very, very well. HPLC is also possible, will be more accurate maybe than the enzyme kit, but more time consuming. So we've looked at some of these kits, and we've looked at the manufacturers of different enzyme kits for gluconic acid, and I'm going to show you some of these results now. So in this slide, this is Chardonnay bunches, and these Chardonnay bunches, again, these were laboratory inoculated, not field. They were mixed, so we had clean, healthy bunches that were then mixed with infected ones, and the weight determined, and from this we got a percentage infection. And we looked at these 3 commercially available kits that are listed on this slide, and you can see the results are comparable. The Thermo Fisher one is not perhaps running parallel with the other 2, but if you put the error bars on, which I've left off of this figure, then they're fairly comparable. One thing to note though is that there is still some gluconic acid in the control clean fruit. So there's some background gluconic acid that's present there. What's my opinion of them? Well, they're great, but hey, look at this figure here. This is looking at gluconic acid, not just in botrytis, but grapes infected with other bunch rot pathogens. And probably the levels are comparable in penicillium and maybe some of the Aspergillus species. But if you've got Aspergillus carbonarius, massive amounts of gluconic acid being produced. So my comment on the kits are, yeah, they're an indicator of fungal spoilage, but maybe there's not great specificity there if we've got a mixture of bunch rotting fungi in the video. And very often you don't. Very often it's just botrytis, it's just one thing. So gluconic acid enzyme kits require medium level of skills, not necessarily specific for botrytis. Analysis time, pretty quick. The assay might take 30 minutes to run, but you might need to do some sample prep beforehand, and maybe applicable for a field situation. I'm not going to say an awful lot about the molecular methods. I will show you just this one graph because we hear a lot about PCR and qPCR, which is quantitative PCR. This is another set of grape bunches infected with botrytis, and we carried out qPCR on these bunches, and you can see it's a good linear relationship. And down at the lower end, we're not picking up botrytis. But the thing to note here are these really large error bars. And this is due to the multinucleate nature of botrytis. And so my opinion of this is that perhaps for botrytis, qPCR there is not great accuracy if we really want a quantitative method. We do need a level of training or skill set in the person who's going to be operating this. So that's a disadvantage. You require some sophisticated equipment, not as expensive as a GC mass spec maybe, but you do need some equipment. Very specific for botrytis, but not perhaps as quantitative as we would like due to this multinucleate nature of the fungus. I'll now go on to more recent work where we've looked at Agostrol. So Agostrol is the fungal sterol that's present in fungal and yeast membranes, and all eukaryotic organisms contain sterol. So animals, humans, grapevines, plants. But the sterols that we contain are different. So we have cholesterol in our membranes typically. Fungi have Agostrol, plants don't have Agostrol. And so Agostrol could potentially be a marker for fungal contamination. And this method has been out there for a number of years, in fact. The method involves extraction of a homogenized solvent, of a homogenized sample using organic solvents, and then subsequent HPLC analysis. Now, we've found that when we grow botrytis, a number of isolates at a set temperature, we know that botrytis mycelium contains roughly 3 milligrams of Agostrol per gram dry weight of fungus. And so using this figure, we can calculate the amount of fungal biomass in a batch of grapes. Now I'm going to quickly talk about some work that we've already published that we conducted back in 2016, 2017. Now the slide that you can see now, you can see 5 batches of grapes. These were Chardonnay grapes that were harvested from a commercial vineyard in the Hunter Valley in 2016. And these grapes were divided into one of the 5 categories based on the level of Botrytis infection. And I know it's not too clear to see from the photos, but that level 0 severity is what appeared to be, based on visual symptoms, apparently clean, healthy grapes. The level 1 is where we had 1 to 3 berries per bunch that were infected. Level 2 was 3 to 10 berries per bunch infected. And level 3 was about 25% of the bunch infected, and 4 was a terrible case. No one would make wine out of that. 50% or more, perhaps, of the bunch infected. And with that number and that knowledge that Botrytis mycelium contains on average 2.97 milligrams of Agostrol per gram dry weight of fungus, we were then able to calculate the fungal biomass in grams dry weight of fungus per kilogram wet weight of grapes. And these were the numbers that we obtained. We were not at all surprised to see that our 0 severity at 0.07 grams dry weight of fungus per kilogram wet weight of grapes, because that's due to the yeast and other saprophytic fungi that might be on the surface of the grape. So here, there's a level of inaccuracy there because of that, because we're picking up other things. And again, lack of specificity, because Aspergillus, Penicillium, all the other fungi also contain Agostrol. However, in this situation, we were pretty confident it was only Botrytis. We couldn't pick up any other bunch rotting fungi when we plated out the material. I'm not going to say much about wine making in this presentation, but this was one study where we did make wine from those grapes that were affected. And I'm not showing you the data on the volatile organic compounds in the grape juice prior to fermentation, but I'm just showing you this slide, which shows the levels of 2 compounds that we are not specific to Botrytis, but we associate with the loss of wine quality, 1-octene-3-ol and 3-octanone. And you can see the sensory perception threshold figures there in blue, and it's really just the 1-octene-3-ol that we're going to look at, which is 1 microgram per litre. And so from this, we can see that these are the 5 batches of grapes. We had none of these compounds in our clean fruit, which was good, confirming there was no Botrytis present. But these levels increase with increasing levels of Botrytis. And you can see here, that's the sensory perception threshold at 1-octene-3-ol. And so from this, we thought perhaps the threshold for contamination is between this level 1 and 2. And some sensory analysis of these wines backed up that assumption. So in this case, we were using Agostrol to try and determine what's the threshold for contamination of wine. And we believe it's somewhere between 0.3 and 1 gram dry weight of fungus per kilogram weight of grapes. The only thing I would say to that is it was 1 vintage, 1 variety, 1 vineyard, and so on. But we have gone on to look at Agostrol analysis a bit further because we think it's a good biomarker of biomass. We can perhaps use it as something to compare the other methods to and work out what weight of fungus we have present there, because that's the issue with a lot of these things. And so in this experiment, we were looking at Chardonnay, Ceylon, and Shiraz grapes. And these were harvested from a commercial vineyard. And you can see again, we've got a reasonably linear relationship there between the Agostrol level and the level of infection. Interestingly, the Shiraz one seems to be higher. That's the SHZ, the purple colour on that graph. Seems to be a bit higher than the other 2. That may be due to the timing of harvest because the Shiraz grapes were harvested later. We harvested them at maturity. So they mature after Cinnamon and Chardonnay. So that could be due to perhaps more background yeast developing on the surface of the berry as the season progressed. But we can see a linear relationship. So what do I think about the Agostrol analysis? Well, you know, we still need some sophisticated equipment and a level of training and skill to carry out this assay. But some wineries do have HPLC equipment running. But this is analysis time of, we could get it down to 2 days as a minimum. It's actually the extraction part and the equipment that we need for the extraction. It takes a bit of time. But ideally, we're going to need 3 days to do the work and then to work out the data. So again, maybe not so applicable for a field situation. It does give an indication of total fungal biomass and is thus not specific for Botrytis. But in those situations with those slides that I showed you for the hunter, you could see it was only Botrytis. But again, there is not 100% specificity for Botrytis scenario. I'm now going to look at the last of the techniques that I'm going to cover in this webinar, and that's the detection of Botrytis antigens. Now, again, this technology has been out for a number of years, probably a decade or more, and I know there have been a number of kits that have been commercialised in the past. There's actually a new kit that's recently been released by a company called Mould. It's currently not available for purchase yet in this region of the world, in the Australasian region. But we've been looking at this kit. It's based on the same technology as the earlier kits. It's the same Botrytis monoclonal antibody, BC12CA4. And the principle is pretty much the same. So the antibody is on a test strip, and this antibody is conjugated to a colour indicator. And so if we have antigens to Botrytis in the sample, they bind to the antibody, and this colour indicator changes colour, and the intensity of the colour it can then be measured and gives us a quantitative indicator of how much Botrytis is present. How this newer kit differs from some of the older ones is that we've got actually a handheld device which allows you to take it to the field. So what's the procedure? Well, you get your grape sample, you homogenise it, and low speed centrifugation. It's just to get out any particulate matter. You could perhaps even just allow it to settle out. And then we place the test strip in a small volume of sample, less than a mil, and you leave it for 10 minutes. This is what it looks like. So here you can see a set of these strips in a series of Eppendorfs, and the sample moves up the test strip by capillary action until it reaches the antigen test line, which I've marked with the arrow there. And if there are Botrytis antigens, they bind to the antibody, and we see this colour reaction. Above this test line, there is a control line, and that's just there to confirm that the kit is working. And if you scan your eyes across this series of strips, you can see the first 3 have clearly got some Botrytis in them. The middle 4 either have no Botrytis or very little, and the remaining 5 in that series do have Botrytis. So how do you read them? Well, this is the cube reader that we use. So this cube reader is 3.5, roughly 3.5 centimetres by 3.5 by 3.5, so it's actually a cube. And then you insert this strip into the cube reader and you read the intensity of the line. So what have we done to evaluate this kit? Well, one of the things that we first carried out was to take Cabernet Sauvignon bunches in the lab, and we inoculated these with a mixture of strains of Botrytis. And so this is lab inoculated, and then we took those bunches, and at daily intervals after inoculation, we measured the Botrytis antigens using that cube reader. And this is the types of results we've got. Now there's 2 curves here, and you'll notice that one is a 1 in 10 dilution, one is a 1 in 50 dilution. And I'll come on to that later, but there is not a strict linear relationship between the antigen and the amount of botrytis. So the dilution that you use is quite important. But you can see day 0, there was no botrytis present. That's the background. Day 1, the botrytis was still starting to grow. And by 2, we were starting to see grey mould symptoms. But as you get to the later stages, day 4, 5, and 6 post-inoculation, now the bunches are covered. The antigen reading is not going up any further, it's plateauing out. And so that's the issue with the kit. If you've got really high levels, then you're not going to discriminate at that higher level of infection. But maybe you don't want to do that, because if you can see it, then you know it's too late, it's too much. In the next series of experiments, we took individual berries. I'll come on to bunches in a moment, but this is individual berries. We inoculated them and then mixed them with clean, uninfected berries. So same as the experiments with the Agostrol and the Gluconic acid. And then we mixed these to get a percentage of infection based on berry weight. And then we assayed the homogenates for the botrytis antigens. And here we examined Chardonnay, Semillon, and Shiraz berries. In these slides, I'm just going to show you the results for Chardonnay, similar results for the other 2 varieties. So this is this 1 in 10 dilution. And you can see down at that lower level, it's a curved relationship. You get this curve, goes up steeply, then plateaus out. And that was the issue with the older kits that were sold here in Australia, that there wasn't that linear relationship, and that was some of the reservations that people had about using them. If we take the same homogenate though, and we dilute it, we're able to flatten out the curve, if you like, and we get more of a linear relationship. So if we go into this 1 in 50 dilution, then perhaps we've got some possibility of using this technology to more accurately predict or to quantify how much botrytis is present. In the next series of experiments, we took lab inoculated bunches now, as opposed to berries. And it's one of these things, whenever I have funding to do botrytis work, there seems to be dry years, so we have to resort to lab inoculated material quite often. And same principle, we mix the bunches to obtain different infection levels. But in this case, we didn't just assay the homogenates for the antigens, we also measured the Agostrol content to try and get an indicator of fungal biomass. And here we looked at the Chardonnay, Semillon, and Shiraz bunches. So this is this 1 in 50 dilution, Chardonnay in blue, Semillon in red, Shiraz in grey on this slide. So again, we're seeing this bit of a curved relationship. So this is this 1 in 50 dilution. So we could perhaps maybe think about repeating this at 1 in 100, which we haven't done. It might flatten that out a bit more. But we can see a semi-linear relationship, I suppose, at this dilution level. This was the results that I showed you earlier, the same sort of bunches with the Agostrol, where we've got a linear type relationship. So the next thing we did was that we compared the Agostrol analysis with the antigens. And when you plot this, we get quite a good linear relationship. The R squared number is 0.9, except if you look at the far left-hand corner at that very low level again. So what do I think of the antigen detection kits? Well, it's simple equipment. It's relatively inexpensive. As I said, the kit we've looked at is not yet available here, but you're probably looking at something of, you know, hundreds of dollars rather than thousands, and the strips are probably, you know, a few dollars each. Once you've skilled your operator, it's a low level of skill training required. The disadvantage is the relationship between the antigen and the botrytis is not completely linear, but maybe this could be overcome by diluting the sample. But the good thing here, two good things, two positives about this technique compared to the others, it's very specific for botrytis. Other fungi, yeasts will not be picked up. And the analysis time is about 20 minutes. So, 5 minutes perhaps to get your sample, homogenize it, spin it down, another 10 minutes to read it. So less than 20 minutes. So if you had to repeat the assay with a dilution, you might be looking at 45 minutes perhaps, less than an hour. But you could do multiple strips at the same time, as I showed in the earlier slide where I showed you the technique being used. So I'm nearing the end of the webinar now, and so I've been through a variety of techniques that we've explored that could be used to detect and quantify botrytis. Many are not specific botrytis, could also detect other fungi. So the question you have to have there is, does that matter? You were just interested in fungal spawnage as opposed to specific botrytis. The costs, the time and equipment skills need to be taken into consideration, and this likely impact on what is it you want to know. Are you concerned about making the wine or just detecting the botrytis? Now this slide is my last one before I move on to my acknowledgements, and I've tried to collate my opinions here and my observations on these different techniques. And these are some of the things that I thought might be important if a grape grower, winemaker wanted to measure botrytis. So the time, I think, is pretty important in a vintage situation where you're getting grapes at the weighbridge coming in. And if you look down that column, the quickest one is the antigen kit, but your walking through the vineyard can also be pretty quick, depends upon the area and size that you want. Specificity, well, the ones I think have the greatest promise for specificity are probably the volatile organic compounds with a targeted analysis for key aroma compounds. The qPCR is specific, and the antigens are also specific. In terms of ability to detect and accuracy, well, the imaging techniques don't tell you what's inside. All the other ones where we're homogenizing the sample, they tend to. In terms of applicability and what could be done now, hyperspectral imaging, well, we could perhaps have a camera as the grapes are coming in or in the vineyard. The VOCs, the volatile organic compounds, not yet, but maybe in the future. Gluconic acid is possible. Agostrol, probably not, because of that 3-day period to processing, but the antigen would be applicable. And then I've got the operator skills there, and that just depends upon the skill set of the person you're doing the work and carrying out. And then finally, the equipment requirements and the cost. And when you get into high-powered analytical equipment, the GC, the mass spec, the HPLC, then you're starting to look at tens of thousands of dollars rather than tens of dollars. And so I'm going to conclude my webinar there. I just want to acknowledge a few people. This is the research team that's fluctuated at times. So the 3 people at the top are 3 postdocs that have been involved with various parts of the research. Michael was my most recent postdoc, and he largely concentrated on the lateral flow devices, the antigens and the molecular methods. Claudia Schumann concentrated on the volatile organic compounds, and Lachlan Schwartz was largely concentrating on the Agostrol analysis, but also the VOCs as well. And the 4 people at the bottom of this acknowledgement slide, John, Lee, Andrew, and Bob, are all co-investigators with me here and academics at Charles Sturt University involved with the work. I just flagged that the kit was provided by MoLogic Limited UK, and that I do thank grape growers who've collaborated with me. And particularly I have to wish to thank Wine Australia. This work has been conducted through various funding phases. The most recent one is a strategic partnership agreement with the NWGIC and Wine Australia. So without Wine Australia's support, none of what I presented would have been possible. And so I'll finish there and thank you for participating. And if you've got any questions, it's question time now, so I'll hand over to Robin. But if your questions aren't answered and you've got something that you really want to discuss with me further, I've got my email address there and I'm happy to talk to anybody after the webinar in the future. So Robin, I'll stop sharing my screen now.
Great, thank you. Thank you very much, Chris. What a great presentation. We've had some good questions coming through. So it sounds like we're not far away from having some really good lab-based methods available, but the field-based methods might be a little bit further away. Sounds like the Botrytis antigen detection method has some really good potential in terms of its specificity, low cost, and the low time required. We've got a question about sample preparation for that method. Do you need to homogenize whole berries, or can you just use juice that's been extracted from maturity samples?
You can use either, and in fact the technique works well even with wine. Some of the work that I did over 10 years ago, I was actually looking at Botrytis Semillon wines from Riverina and Sauterne wines from France, and assaying the level of Botrytis antigens in those wines. So you can use juice, you can use homogenized sample, you can use anything basically. If you go to say like a dessert, Noble Rock dessert wine, huge amount of Botrytis antigens there. You have to dilute 1 in 100 or much more.
Okay. All right. So with the I'll use my Australian pronunciation, Agostrol detection method. It sounds like you've got some really good threshold information for the contamination of Botrytis. With the Botrytis antigen detection method, you didn't mention any threshold information there, but then you compared it with the results that you got for the Agostrol results. So it sounds like around that 5% Botrytis severity level, your results showed that that's when people could pick up Botrytis in the sample. So do you feel confident that you've got a threshold level for that Botrytis antigen method?
No, I don't, because the work with the Agostrol and the Chardonnay from 2016 was just Chardonnay, and it was just one vineyard, and it was actually quite a broad range. So the next step, I think, in this research, if it goes forward, it would be to look at that winemaking and actually do some sensory analysis around it. So that I would think is the next step. And the other thing is that the threshold will depend upon the grapes and the variety and all sorts of other things, and also the winemaking as well, and techniques the winemaker might do in terms of fining agents to pull out off flavours and things like that.
Okay, thank you. So we've got a question about the pros and cons of an electronic nose for VOCs. So you talked a little bit about mobile devices that might be available in the future. Can you talk a little bit more about this concept?
Yeah, look, I think this is probably the way of the future. What we would really like to have is some sort of simple handheld device that would selectively pick up these compounds in a vineyard situation in a weighbridge, but within the current funding that we've had in the current project, we haven't got that far yet. but you know, perhaps come back and talk to us in 3-5 years' time, and then we might have things like that sort of thing that we can wheel out.
Okay. You talked about the Botrytis antigen detection method not being particularly or plateauing out when you got to high levels of botrytis. There's a question about what the upper limit of botrytis infection is for that method.
Well, really, there's no upper limit, because you just dilute the sample and you bring the curve down till you've actually got that linear graph. I was going to show, but I don't think it was relevant for this webinar, some data that I've got on the dessert wines where it goes up really, really steeply. But that's what you'd have to do. The thing is, you don't know what's there till you do the reading. But it's a bit like measuring the absorbance of something in the spectrophotometer. You shouldn't measure the absorbance when it's greater than 1 because of Beer-Lambert's law. You need to dilute your sample. So with gluconic acid enzyme assay, if you're getting readings of above 1, then you have to dilute it. It's the same sort of principle, really.
Okay. All right. Thank you. So we've got another one more about the FDIR analysis method. And it's a question about the specificity of that method. So the person has said, I think we need some fitting models suitable for each environment, types of mould, weighting, etc. What are these in general, or how can we find out the model we have to use?
Yeah, I think we've pretty much abandoned FDIR because I could see the limitations in terms of, yes, we could discriminate between berries, but if we wanted to go a step further, you know, whole bunches, it wouldn't work. And similarly, with all the data handling and analysis, it's just not going to be appropriate for a winery type situation and when you want quick answers. So more work could be done in that area, but for us, that avenue of research is actually stopped.
Okay. There's been another question about the use of using the antigen kit on a soil sample.
Yep, theoretically, it could work. You know, it's very specific for botrytis. I don't see any reason if you had botrytis spores or mycelium in the soil, if you wanted to measure that, I think you could. I don't know why you would do it though, because undoubtedly botrytis is present on the debris in the soil and it's there all the time. It's really when the conditions are right and you're getting transmission to the vine that you really want to pick it up. So yes, I don't see any reason why you couldn't measure it in a soil sample. I would envisage that you would take a soil sample and perhaps shake it up in buffer, let it settle out, and then, you know, assay the supernatant. So I don't see why it wouldn't work, but not something I've tried.
All right, thank you, Chris. And just one more question. Can you please give us your thoughts on the lacase tests that are already commercially available for measuring botrytis? You mentioned it briefly in one slide, but you didn't go on to add it in the comparison table at the end.
Yeah, I think with lacase, the thing with lacase, why does the fungus produce lacase? That's the first question to ask. The fungus produces lacase along with the other cell wall degrading enzymes that it produces when it's infecting grapes. Now, if you've got berries that are split and damaged, it's going to produce less lacase. It's an inducible enzyme. So it's the variability in the amount of lacase that the botrytis produce. Now, why are we interested in lacase? It's because of the spoilage of wine. So if you want to look at the potential for spoilage, then I think lacase is a valid method. But if you're trying to say, do I have botrytis present or not? I don't think so. Because you can get some isolates, you can grow up botrytis, it doesn't produce any lacase. It's an inducible enzyme. It's induced by copper, for instance, copper inducible enzyme. So I know, perhaps if you spray your vineyard with copper, then you might get more lacase producing botrytis. I don't know, but there's all sorts of things like that I've speculated on with regards to lacase. So for me, I'll be uncomfortable using lacase as an indicator of botrytis, an indicator of potential wine spoilage. If you've got a must where there's lacase, when you make your wine, it is going to go brown.
All right, thank you. Very good. So unless there's any other questions, thank you, Chris. It was, a great presentation, really insightful. I think we've got some good things on the horizon, but obviously a little bit more work to do to get those thresholds and the methods suitable for use in a vineyard setting. I'm sure everyone who attended today got a lot out of your presentation, so thank you. I'd also like to thank the audience for joining in and taking part, particularly those who asked the questions. It's always good to get your questions. I'd also like to remind you that the presentation has been recorded and will be made available to you on the AWRI's YouTube channel. So a link will be sent out via email. I'd also like to acknowledge Wine Australia for providing funding and support for webinars via the AWRI Extension Project. So our next webinar is on Thursday, the 29th of April. It's by Dr. Anthony Borneman from the Australian Wine Research Institute, and he will present the origin of Chardonnay clones with historical significance in Australia and California. So if you'd like to register for this session, please visit the AWRI website. Thank you, and we look forward to seeing you again. Thanks again, Chris.
Thank you.